and Conservation Biology seminar series today on Friday the 13th. This is sponsored by the Fisheries and Wildlife, Forest Ecosystems and Society, Integrative Biology, Botany and Plant Pathology, OSU Research Office, and the Forest Biodiversity Research Network. This term, our theme has been how can ecology, evolution, and conservation biology contribute to our understanding of global pandemics. And to contribute to this discussion today, we have Dr. Mordecai. Dr. Mordecai has a love for empirical data and modeling and has found a way to unite that interest with ecology in a meaningful way by studying the ecology of infectious diseases. She is currently an assistant professor in biology at Stanford University and has been there since 2015. She got her Bachelor's of Science at the University of Georgia, got her PhD from the University of California, Santa Barbara, and was an NSF postdoctoral research fellow at the University of North Carolina at Chapel Hill and North Carolina State University. And with that, I want to thank you all for joining us, and I'll turn it over to Dr. Mordecai.
Thank you very much, Jessica. Let me get my slides shared here. All right, does that look good? I'm assuming yes. Okay, thank you so much for the opportunity to be here. I'm really excited to be able to share with you some of the research my lab has been doing over the last 6 years since I've been at Stanford. Broadly, our research is about how global change affects the ecology of vector-borne disease. And before I get started, I would like to acknowledge the really wonderful people who have joined my lab over the last 6 years, including a wide range of different grad students and postdocs and lab technicians. And without them, this work really would not have been possible. I also want to acknowledge the funding sources that have made this work possible, including NSF EID grants, NIH R35 grants, and then several centers and institutes at Stanford, the Center for Innovation and Global Health, the Woods Institute for the Environment, the King Center for Global Development, and the Jasper Ridge Biological Preserve. This is a photo showing some of our field research on plant pathogen interactions in Jasper Ridge. So for many of you who may not be disease ecologists or may not think about parasites all the time until this past quarter, one of the reasons that I find parasitism so interesting and so cool is because it's really ancient and it's really ubiquitous. For hundreds of millions of years, parasites have been evolving to be able to attack their hosts, to not only attack them, but to penetrate their defensive barriers, to steal resources from their hosts, to attach themselves and really successfully exploit their hosts, and to transmit from one host to the next. So a beautiful example of this is the scolex or mouth parts that you see, the really hooked part of this tapeworm here under electron microscopy. And parasitism has had really diverse and extensive ecological and evolutionary impacts throughout the history of life on Earth. You can think about parasites as having individual level fitness effects on their hosts, which can then translate into effects on population dynamics, effects on evolutionary trajectories, effects on ecosystem services and the flow of energy and nutrients through ecosystems and so on. So there's this really kind of long and rich history and diversity of ways that parasites are important in the world. And of course, not least of this by any means is the impact that parasites have on humans. Of course, the major burden of diseases in humans that are caused by parasites, pathogens, viruses, bacteria, and so on. Of course, the major context for all of this work is that humans are really rapidly changing the Earth. Global climate change is probably the most well-known example, but we're also really rapidly changing land use on Earth. The Earth is urbanizing. Humans are moving around the world incredibly fast. We're migrating, we're changing ecosystems, we're bringing new species into new places, and we're driving other species extinct. And all of this has led to this kind of doom and gloom scenario emerging from a lot of the disease ecology literature, suggesting that a warmer world is likely to be a sicker world. And this is really exemplified by the malaria literature. This is just one example from a whole body of research in malaria literature, suggesting that future climate change is going to have a really big and bad impact on malaria, especially throughout sub-Saharan Africa. This is just a model projecting the change in malaria transmission by 2050 under climate warming. And what you can see from this map is that almost all of sub-Saharan Africa is projected to get much, much worse for malaria transmission, besides a little bit of the fringe region here along the Sahel where it's getting too dry. But in contrast to that warmer world is sicker world idea, research in my lab and in other disease ecology labs has really embraced the idea that parasites are embedded within ecological communities. So it's not simply one host and one parasite. We have parasites that interact with a variety of other parasites and microbes within their host individuals and within their host populations. We have hosts that interact with other host species and non-host species, as well as vectors. And we have multiple dimensions of the environment, including the climate, as well as the habitat structure and connectivity. And these environmental features are affecting both the parasites and the hosts and vectors. And so what emerges from this picture is a really complex potential system of infectious disease, where each one of these arrows can represent an interaction that could potentially be nonlinear, or the emergent effect of multiple arrows could in turn introduce additional nonlinearity. So all of this points to the idea that this simplistic idea that a warmer world is a sicker world may be an oversimplification. And there may be much more nuance to this story about how global change affects infectious disease dynamics. And so that's really the central goal of my research program, is to understand how global change is altering infectious disease dynamics, and to really get at, can we predict these changes, and can we explore that nuance, that nonlinearity, by making this complexity also be predictable? And the research I'm going to present today focuses on vector-borne disease. These are diseases or pathogens that are transmitted by biting arthropods like mosquitoes, ticks, flies, and fleas. And these are really interesting, of course, because they include things like malaria and dengue and yellow fever, some of the globally most important human vector-borne diseases. They're also really interesting from an ecological standpoint because these pathogens have to pass through this arthropod vector stage. And in fact, for parasites like malaria, that's where the sexual reproduction of the parasite actually takes place. But this little insect or this little arthropod is like a microcosm of the environment around it. It's affected by temperature and other aspects of climate like precipitation and habitat and movement around the world. So to address this big picture question about how global change will affect the spread of vector-borne diseases, I'm going to divide this talk into two parts, where the bulk of the talk, about 2/3 of it, will be about climate change and vector-borne disease. And in the second part, I'll give a little vignette talking about how land use change can also affect vector-borne disease. So narrowing in now from big picture effects of global change on vector-borne diseases as a whole, how will climate change specifically affect vector-borne disease transmission? And this is work that I've been doing along with postdocs Marta Shockett and Jamie Caldwell and PhD student Nicole Nova in the lab. So to understand how vector-borne disease transmission works, we have to think about the life cycle of the vector, in this case, a mosquito. This life cycle, as most of you probably know, involves an aquatic stage. So these adult female mosquitoes lay their eggs in a body of water of varying sizes, depending on the ecology of the specific mosquito. It could be something like an open pond or the sides of a riverbank, or it could be something as small as a bottle cap or a bucket around the household. Once these eggs are laid, they hatch into these larval stages that go through 4 different larval stages as they grow and molt. And then eventually they pupate and they emerge as adults. These adults then go on to bite hosts, including humans, depending on the mosquito species. And the disease transmission cycle basically hijacks this mosquito life history. So it's only the female mosquito that bites vertebrates and takes blood meals. And with those blood meals, the female is producing new eggs. And in the process, if the parasite or virus is contained within that blood meal, the mosquito is not just like a flying syringe. It's not just taking parasites from one person and putting them into another person. Instead, there's this really exquisite biological process that goes on within the mosquito, where the parasite has to get out of the blood meal and not get digested. It has to break through the midgut barrier, replicate and disseminate throughout the body, and then finally bind to the salivary glands, where it gets injected into the next host. And this process, which is called the extrinsic incubation cycle, can take days to weeks, depending on the temperature. And different vectors have different levels of competence or ability to acquire and transmit a given parasite. So given all this, we expect lots of these traits of mosquitoes to be temperature sensitive because they're taking place within the body of a small ectotherm. But not only that, we have a pretty strong expectation for what that temperature sensitivity should look like. We know this from really a wide range of literature in the ectotherm physiology research area, including a body of theory that's built from first principles about enzyme kinetics, as well as a really broad body of empirical research showing nonlinear relationships between temperature and a variety of different traits in many different taxa. So what I mean here is if you have the performance of a given trait, whether it's a development rate or a metabolic rate or survival or reproduction, we often see this nonlinear relationship between the performance of that trait and temperature. So we have a critical thermal minimum and a critical thermal maximum temperature at which the trait goes to 0. And then we have this nonlinear relationship in between, where there's an optimal temperature somewhere in the middle. And although this is one of the most well-established processes pretty much in all of ecology and certainly in biology as well, these types of nonlinear thermal response models have often not been incorporated into vector-borne disease transmission models. And in particular, these models that suggest that a warmer world is a sicker world often rely on assumptions that temperature linearly or monotonically increases the performance of these different traits. And so the goal of our research, first of all, in this first part, is to reconcile the disconnect between what we know from ectotherm physiology theory and empirical work and what vector-borne disease models have traditionally assumed about how temperature affects transmission. So our approach, as I mentioned, is to simply reconcile this disconnect. So we're going into the literature and we're looking for experimental lab studies where entomologists have taken mosquito populations and held them at different constant temperatures and measured these important traits that are involved in disease transmission, like biting rate, transmission and infection probability, mortality rate, parasite development rate within the mosquito, and mosquito abundance. So we can take these trait measurements, these empirical measurements, and then we can fit mathematical models to them to describe the relationship between temperature and trait performance. Then, taking those individual trait thermal responses, we can combine them into an overall model for transmission versus temperature, which we can express as R0. Many of you, probably because of COVID, have been hearing about R0 recently. It's the number of infected individuals that are expected to arise from a single infectious individual introduced into a fully susceptible population. And what we're focused on in this research is not so much the absolute number of how many secondary cases we get, but the relative number. So where is R0 maximized with respect to temperature? And this is a model that's been developed and refined over 120 years now, since the discovery of mosquito transmission of malaria. And what I want you to notice from this model equation, which again, we derived from the literature, is that it's a nonlinear function of many traits that are mostly traits of the mosquito or parasite within the mosquito. So as I mentioned earlier, there's lots of opportunities for nonlinearity to come into play here, both because the individual traits are nonlinear and because they combine in this nonlinear way to affect R0. So we're going to use R0 as our common currency for measuring how temperature affects the overall transmission process by bringing together all of its individual effects on the different traits. Then, because these R0 models are parameterized using empirical data from the lab, we're then very interested in comparing them with patterns of transmission in the field, because we really want to know, do these lab-based experiments actually hold up in these much more complex and dynamic systems occurring in the field? So we're going to try to compare our predicted R0 versus temperature with observations in the field. And then finally, we can use those validated models to project current and future transmission risk as a function of temperature. So to narrow in this question a bit more, we now want to ask, how does temperature affect malaria transmission specifically? And this is work that I began as a graduate student. What we found here is that physiological responses of the malaria mosquito to temperature are hump-shaped in exactly the way that the theory and the previous empirical work had predicted. So this is an example for the development rate from egg to adult, showing this hump-shaped relationship with a peak around 28 degrees Celsius and critical thermal minima and maxima around 16 and 34 degrees Celsius. And in fact, if we look across the entire suite of traits that influences malaria transmission, we see strong support for nonlinear responses. Although in some cases, as you can see, the data are a bit sparse, even for malaria, which is arguably the best studied vector-borne disease. When we now take all these nonlinear trait thermal responses and incorporate them into a temperature-dependent R0, we're predicting that this also has a hump-shaped relationship with temperature. For falciparum malaria, this is the most important human malaria parasite in most of Africa. And what we're predicting is that this relationship peaks at about 25 degrees Celsius. Now we can contrast that with the previous models that had used the linear thermal response functions. And those models predicted about a 7-degree warmer thermal optimum of about 32 degrees Celsius. So for comparison, 25 degrees Celsius is about 78 degrees Fahrenheit. So this is a fairly cool optimal temperature that we're predicting for malaria, considering it's a tropical disease, or at least it is today. So we were really interested in validating this model now by comparing it with patterns of transmission in the field. So what we did to validate the model is we were lucky enough to have access to a publicly available data set on entomological risk of transmission. And so we plotted this entomological risk, which is the number of infectious mosquito bites per person per year, calculated across locations in Africa over 30 years. We plotted that against the average transmission season temperature. And what we see here is, first of all, there's quite a bit of variation that's not explained by temperature, as you might expect, because there are things like bed net usage and housing construction and rainfall and humidity and other factors that also drive malaria incidence. But at the same time, we can see that there's a clear temperature-dependent relationship where the highest levels of entomological risk are occurring right around that predicted optimal temperature of 25 degrees Celsius. So to zero in as best we could on where we think the variation is most likely to be driven by temperature, we pooled these points by temperature. So we binned them. So there were 10 points in each bin, and we're just going to highlight the maximum within each bin. And these are the places where we expect that other factors are the least limiting, and therefore temperature may potentially have the biggest influence on transmission. So if we now look at those maximum points, which are now the filled circles here, you can see that, again, these maxima tend to increase as we get closer to 25 degrees Celsius, and they tend to really steeply decline as we get above 28 degrees Celsius, supporting this prediction, again, that from this mechanistic lab-based model, that malaria transmission peaks at 25 degrees Celsius. Now, this work was all done when I was a graduate student, and I guess 6 or 7 years later, when I came to Stanford, I was able to finally test this prediction not only in entomological risk, but also in human infection data. So working with a collaborator, Dr. Desiree Lebeau in the Stanford Med School, we were able to have access to a study that she did across over 2,000 children across households in Kenya, where they were able to match up measurements of human malaria incidence in children with the average temperature during that transmission season. And we see, again, this really striking pattern where the highest levels of incidence were occurring right around 25 degrees Celsius. And as the temperature increased above 25, we saw really rapidly declining rates of malaria incidence in children, which allows us to sort of close the loop from the lab-based physiological studies of individual mosquitoes to population level projections of how mosquito transmission responds to temperature, and finally all the way to human incidence. So what are the implications of this for climate change? In contrast to that warmer is sicker prediction, what we're predicting from this model is that warming may speed transmission in some of these cooler places that are below 25 degrees on average. But at the same time, warming may actually slow transmission in some of the warmer places. Of course, one of the big unknowns that we have here is what is the relative role of temperature compared to other factors, and how much potential is there for the mosquito and the parasite to adapt to warming temperatures. But given what we know right now, at the very least, we can say warming is not expected to expand malaria transmission in places that are already at or above 25 degrees. So the next thing we wanted to do was to understand how broadly this situation applies across a wide range of different mosquito-borne diseases. So I was interested in, had there been similar sort of restrictions in the previous models of other mosquito-borne diseases, and can we compare and contrast across different mosquitoes and different parasites where their thermal optima and their thermal limits are, and therefore what's the potential for climate change to impact them? So this work last year was really excitingly was published and was the cover story in Ecology Letters last October. So again, we're applying this exact same approach that we applied for malaria, but now we're applying it to these 11 different mosquito-borne diseases that are some of the most important human mosquito-borne diseases. Things like dengue, chikungunya, Zika, West Nile, Ross River virus in Australia, Rift Valley fever virus, Eastern equine encephalitis. If you're from the East Coast, you may have heard of that one, triple E. And what we find looking across this whole range of different vector and parasite species is really strong support for these nonlinear thermal responses. So again, this is the mosquito development rate trait shown for Culex annularostris, which is an important Australian mosquito. But what we see is support for this across all the different traits that are relevant for transmission and across all the most important human vectors of disease, including Aedes aegypti and Aedes albopictus, which are the dengue vectors, Culex pipiens, tarsalis, and Quincke fasciatus, the West Nile vectors, and Anopheles gambii and Stevensii, the malaria vectors. So we've been publishing this in a series of papers. And what we wanted to do with this new synthesis paper was to combine the temperature-dependent thermal responses of the different traits and of R0 itself. So what we were able to show in this paper, first of all, is that there's really broad consistency across different taxa. So what I'm showing here is 5 different traits related to mosquito life history, like development rates, fecundity, biting rate, and survival. And each of the different colored lines represents a different mosquito. We're just showing in the legend the different mosquitoes and the pathogens they transmit. You'll notice in a couple of cases, there are some dashed lines representing cases where we had more than one trait measured from the same mosquito. What we see very consistently here is that all of these relationships are hump-shaped, and yet there's quite a bit of variation across the different mosquito species, particularly for some of the traits like survival and fecundity. There's a lot of variation in where the optimal temperatures are and where the thermal limits are. And we see the same type of thing when we look at the infection and transmission related traits. We see a lot of variation across these different species. So when we put all these temperature dependent traits together and calculate a temperature dependent R0 for each of these systems shown in the different colors, we can see a range of different relationships between transmission and temperature. The first thing to notice is the consistency, that they're all hump shaped. They all have lower and upper thermal limits, and they all have optimal temperatures. And all of those optimal temperatures range from about 24 to 29 degrees Celsius. So dengue and Zika, transmitted by Aedes aegypti, have some of the highest thermal optima. Ross River virus, that important disease in Australia, is in the middle, along with malaria. West Nile and Triple E, which are the more temperately distributed pathogens, or diseases, have more temperate thermal optima as well, between about 23 and 24. We can also see that there's quite a bit of variation, especially in the lower thermal limits for these diseases. We can see that anywhere from 10 to above 20 degrees is where these different transmission curves go to 0, whereas there's a bit less variation across systems in the upper thermal limits. We can see that there's some support for some what look like pretty general upper thermal constraints on transmission above 32 to 35 degrees Celsius. So how will climate change affect the spread of vector-borne diseases? I think it's safe to say at the low temperatures from about 12 to 24 degrees, any increase in temperature is likely to increase transmission of almost all of these diseases. Likewise, once we get above 29 degrees Celsius, extremely warm temperatures, any further warming is only going to decrease the potential for transmission of all these diseases. But what's really interesting is this area in the middle between about 24 and 29 degrees Celsius, because these are the temperatures where most places have their spring and summertime climates. And these are the temperatures at which some pathogens are really, really steeply declining. Others are steeply increasing, and others are either not responding very much to temperature at all in that range, or they're going over their turnover point. So to focus in on 2 of the most important ones, malaria and dengue, I just want to show what this looks like more specifically on a map for the future of temperature-driven transmission risk. So in sub-Saharan Africa, what I'm showing here is a hotspot of malaria climate suitability. So remember that malaria thermal optimum is 25 degrees Celsius. And what we're projecting is that the hotspot, where there's lots of people exposed to highly suitable temperatures for many months a year, is currently in coastal West Africa. But as the climate continues to warm, we expect to see this hotspot shifting and moving towards up the higher elevation region in the Albertine Rift region in East Africa. So this is a place where the temperatures are currently a little bit too cool for optimal transmission. But by 2080, with unmitigated climate change, we expect to see malaria spreading up there. And at the same time, in much of West and Central Africa, the temperatures are becoming too warm for optimal malaria transmission. But at the same time, because dengue transmission has a 29 degrees Celsius optimum, in contrast to malaria, we now expect the hotspot, which is kind of starting out in the same place, to be broadly expanding across sub-Saharan Africa. So this is a really important message for public health, because first of all, malaria, of course, is very important, and malaria control efforts are very important and have been very successful over the last 20 years. But it's important to recognize that some of the success may actually be coming from decreasing climate-driven suitability, and that we're going to see some expansions in malaria transmission suitability, especially in high elevations. But at the same time, we should acknowledge this expanding risk for dengue transmission, because it's transmitted by the Aedes aegypti mosquito, which is a container breeding domestic mosquito that bites during the day instead of the night. So things like insecticide treated bed nets that are so effective against malaria don't work against the dengue mosquito. Of course, malaria therapeutics and malaria diagnostics also don't work against dengue, which is a virus. So it's very important, while we continue to maintain public health efforts towards managing malaria, we have to recognize the potential for this expanding threat of dengue and other viruses transmitted by that mosquito, and retool our public health efforts accordingly, especially since the vast majority of people on the African continent may currently be susceptible to these viruses and haven't been exposed previously. So the next thing we wanted to ask with this research is, how well can climate-driven models actually predict the risk of disease that we see in the field? And the challenges here are data availability and biases, of course. We don't always have great measurements of transmission in the field. There's often spatial aggregation, so things are reported at the state or country level. There's variation in climate and in disease over time. There are time lags, which themselves depend on temperature. And there are multiple drivers going on at the same time. So it can be really difficult to separate out causal effects of climate variation on disease transmission. And for that reason, we've taken some somewhat indirect approaches in the past, as I'm showing here in our example for malaria. Another example of ways that we've gone about this problem are for West Nile virus. This is Marta Shockett's work that was recently published, where our temperature-dependent R0 models for the 4 important vectors across North America all peak somewhere around 24 to 25 degrees Celsius. And what Marta found when she looked at the average incidence of West Nile across US counties is that incidence, although it's quite messy, again, there is some predictability in this incidence that's attributable to climate, where the average summertime temperatures around 24 to 25 degrees Celsius, those were the counties that had the highest levels of human incidence of West Nile disease, suggesting, first of all, that there is a detectable signal of temperature effects on West Nile transmission, and it's nonlinear, and the peak is occurring right around the estimated temperature-dependent peak of R0. The next thing that we're able to do with this type of mechanistic model that's really cool is it actually helps us to reconcile and make predictable some previously idiosyncratic results that were in the literature. So this is work by Jamie Caldwell, where she reviewed previous field studies that had looked at relationships between dengue and temperature. And she recorded the magnitude and direction of the inferred effects of temperature on dengue transmission. And then she plotted that according to the average temperature in the study system. So what we see here with these positive arrows is that there's a lot of places where temperature has positive effects on dengue transmission, but they're primarily at the portion of the curve where we're rapidly accelerating in R0. Whereas if we look at the temperature range from about 25 to 32 degrees Celsius, as we would expect, there's very minimal effects of temperature on transmission in the field in this temperature range, because again, we're right around the peak. But then we start to see actually negative effects of temperature on dengue transmission in the field when we get to temperatures that are as warm as 33 and above. So again, this helps us to reconcile something that was previously a little bit mysterious in the literature. Why do we see so many different types of effects of temperature on dengue? It's because of this nonlinear relationship with transmission. Now, Jamie further wanted to ask, not only looking spatially, but can we actually use variation over time in climate dynamics to predict epidemic dynamics? How well can we actually predict epidemic dynamics from just knowing about the climate? Because if we could do that, that would be really useful for early warning and forecasting systems. So what Jamie did was she took advantage of collaborations we had with people in Ecuador and Kenya, where they had been sampling mosquitoes and human infection incidents across climate gradients for dengue, chikungunya, and Zika and their vector. And they had also been measuring temperature, humidity, and rainfall. And Jamie developed a dynamic version of our model, which used the same temperature-dependent traits that I showed you earlier for the R0 work. But now, instead of an R0 model, she's using a more complicated model that tracks the movement of mosquitoes and humans through susceptible, exposed, and infectious, and for humans, recovered classes. So the details of the model aren't important right now, but the point is that we're feeding in these dynamic climate measurements, and we're feeding out predictions of mosquitoes and disease cases over time. And then we're able to compare those with what we actually observed in those 4 locations in Kenya and 4 locations in Ecuador. And what Jamie found, which was really exciting, is that these climate-driven models were actually able to do a really good job of predicting the number of epidemics we saw, the timing of those epidemics during the season, and the duration of the epidemics. So R-squareds here are 0.51 to 0.79, just using observed climate dynamics fed into this dynamic model. And this applies to both locations in Ecuador, which are shown in circles, and the locations in Kenya, which are shown in triangles. Interestingly, though, although we could predict the timing and occurrence of these outbreaks, the model wasn't able to do a good job of predicting the size of those outbreaks, so how many people actually got infected. And what we hypothesized here was that that's because the size of the outbreak is not as much driven by climate suitability as it is driven by the number of susceptible people that are available to be infected. And so to test that hypothesis, we then worked, this is work with graduate student Nicole Nova, to ask how climate and host susceptibility combine to affect dengue transmission dynamics. So this is building on a new sort of emerging, I guess it's not that new, but an emerging approach spearheaded by George Sugihara and colleagues called empirical dynamic modeling. And I'm not going to go into the details of how this works, but the basic principle is that you can reconstruct a state space. So for example, if X is temperature and Y is the number of susceptible people and Z is the number of dengue cases, we can track our system as it moves through time, through temperature, number of susceptibles, and dengue cases. And we can reconstruct what's called this attractor. And we can use that attractor mathematically to examine causal relationships between these three variables, as well as to do prediction going forward. So if we know what the system has looked like in the past, we can use that reconstructed system to project its future. And the reason we had this opportunity to do this is because we had this amazing data set available from the CDC through their dengue forecasting challenge. This is weekly dengue incidents from San Juan, Puerto Rico, from 1990 up to 2010, and actually even beyond that. So this is showing incidence over time in the top panel. They also provided data on temperature in red, rainfall in blue. And then using this empirical dynamic modeling framework, we were able to infer an index of the number of susceptible people over time. And what we find when we applied empirical dynamic modeling to this system is that dengue dynamics are highly predictable, first of all. So when we build this attractor, we're able to predict within sample with a correlation coefficient of about 0.75, using only the information about climate and susceptibility. And out of sample, we actually did even better. In the 4 out of sample years, the correlation was about 0.95. What's interesting, though, is that this actually gives us some more causal understanding for how climate and susceptibility are combining to allow us to predict dengue dynamics. So here, looking at the panel on the right first, this is our temperature-dependent R0 model. And what we would predict, if we look at the slope of R0 with respect to temperature, we expect to see the highest slope, the maximum rate of change, occurring around 25 degrees here. We expect to see a shallower slope occurring around 29 and around 22 because of the nonlinearity of the model. We also expect that, as I mentioned our hypothesis earlier, we're only going to see really strong effects of climate when there are enough susceptible hosts available to be infected. So what we're able to find using this empirical dynamic modeling framework is that there's this threshold for host susceptibility. So when our index of host susceptibility was low, temperature basically had no effect on transmission. So I'm showing temperature here on the x-axis and the change in incidence with respect to temperature. So that slope on the y-axis. And we can see that for most of the points on this reconstructed surface, when there's low host susceptibility, we see basically no effect of temperature on incidence. But by contrast, once our susceptibility gets high enough, we start to see mainly positive effects of temperature on transmission, on incidence, when we're between 24 and 26 degrees, and those slopes start to decline towards 0 and even go below 0 at 28. Now, we didn't have many observations above the predicted thermal optimum, so we weren't able to test evidence for that. But this supports the idea that host susceptibility is modulating the effect of temperature on dengue transmission. So to summarize this first part, the effects of temperature on mosquito-borne disease transmission are, first of all, highly nonlinear. The individual traits that drive transmission respond nonlinearly to temperature. And not only do those individual traits respond nonlinearly, but they differ across different vectors and different pathogens. So you may not have caught this subtlety, but when I showed all the different temperature-dependent R0 curves together, in some cases, it's the same pathogen transmitted by different mosquitoes, and they have different thermal responses. So these thermal optima and thermal limits vary, which is why it's really important for empirical research to continue to investigate these thermal responses so we can understand better how does temperature affect the transmission of each given disease. So what we can expect for climate change is that because the relationship between temperature and transmission is so nonlinear, we expect to see a lot of shifts in climate suitability rather than widespread expansions in transmission. So rather than the warmer is sicker idea, we're often going to see new diseases appearing in new places, or within an existing place, we may see shifting seasonality or shifting geography of where a disease is occurring. And we may see new vectors and new diseases becoming important. So we may see shifts, for example, from more malaria transmission to more dengue transmission in a given location. But the most important caveat here is that all of this is going to be mediated by human activities. So we're only going to see these climate-driven shifts in places where the opportunity is there. And that's where the second part comes in. So in this shorter second part of the talk, I want to investigate effects of land use change on vector-borne disease. And specifically, this case study is about how deforestation affects malaria in the Brazilian Amazon. This is work done by Andy McDonald when he was a postdoc in my lab, and it was recently published last year. So in the Amazon, in contrast to what I expected before I started this project and what you may have expected, when you think about deforestation, you might think about huge swaths of forest being cleared and no more trees remaining. But actually, especially in the beginning stages of deforestation, it often looks a lot more like this. This is what's called a fishbone pattern of deforestation, where you get a major highway going through the forest, and that provides access. So you get these little offshoot roads. And along those roads, you see really major deforestation coming right up against the edge of the forest. And what this type of deforestation tends to do is it maximizes the amount of forest edge, and it brings people up against that forest edge where they're living and working. Now, in the Brazilian Amazon, malaria is transmitted by Anopheles darlingi, which is a forest edge associated mosquito. And it's primarily caused by two parasite species, Plasmodium vivax and Plasmodium falciparum. So this forest edge mosquito breeds in natural pools of water, which tend to accumulate, especially in places where there's been a lot of disturbance, like clear cutting or mining. And what we've seen over time is that although historically levels of malaria were much higher, by the 1960s, malaria had really gotten under control. So in black is the total malaria, in red is vivax, and in blue is falciparum malaria, so two different parasites. But after the 1960s, we saw this really dramatic resurgence in malaria cases. And this coincided in time just a few years after the Trans-Amazonian Highway was built. And more generally, the Brazilian government at this time was really promoting development of the Amazon and incursion into the Amazon, settlement in the Amazon for economic reasons. So what we saw in time was, policies that promoted settlement of the Amazon followed by a huge resurgence in malaria, suggesting that there could have been some role of deforestation on malaria. If we look spatially, this is now a map of all the municipalities in the Brazilian Amazon. These are some of the data that are going to go into what I'm showing next. We can see malaria, falciparum malaria in the left panel, the rate of deforestation per year in the middle panel, and the remaining forest cover in the right panel. So if you look at these, although you may see some spatial associations, especially if you squint really hard, it's not totally clear what the relationship is just by looking spatially. And even if there is a spatial relationship, it's not clear that that's really a causal relationship. So this is really the question that we wanted to address, is what is this causal relationship between malaria and deforestation? And what we found in the literature is that the previous evidence was mixed. In some cases, we saw a strong positive effect of deforestation on malaria. In other cases, we saw no effect. And in some cases, we even saw the opposite, where places where there was more forest conservation was where we saw more malaria incidents. And a lot of this varied by location and by study type. So what we were interested in doing is asking, is deforestation increasing malaria incidence in the Amazon broadly across the entire Brazilian Amazon? So we wanted to develop a systematic estimate for the effect of deforestation on malaria. And so Andy's hypothesis here was that deforestation increases malaria incidence by creating that forest edge and bringing people up against the forest edge. But he also hypothesized that there was potentially this negative feedback of malaria risk on deforestation, because when there's really high malaria incidence, people might be too sick to work, or they might be changing their land use practices, they might be abandoning settlements, and so on. So he hypothesized this socio-ecological feedback between deforestation and malaria. The other hypothesis that Andy had is that this relationship between deforestation and malaria would vary across land use gradients. So here we've adapted a figure from Jonathan Foley's work, which just shows stages of land use transitions. So we start out in pre-settlement with natural ecosystems, and then we get some frontier clearings, some subsistence agriculture and small-scale farms, followed by more intensive agriculture, and eventually urbanization. And what Andy hypothesized here, based on our reading of the previous literature, is that the peak of malaria incidence and its association with deforestation would really occur in this frontier and subsistence agriculture stage. So in the interior of the Brazilian Amazon, it's dominated by small and medium-sized landholders. That these are the places where people are getting a small patch of land, they're farming it themselves, they're clearing it themselves, they're living in kind of small, unimproved housing situations, potentially with not good protection against malaria mosquitoes, and basically living and working right on the forest edge. By contrast, in the outer parts of the Amazon, most of the forest is already gone. And most of what we're seeing here is intensified agriculture with large landholders that predominate, and people are living in better housing conditions, and the forest edge has been pushed further away from where people live and work. So we hypothesize that the relationship between deforestation and malaria varies across land use and is strongest in these frontier regions. So to answer this question across the entire Brazilian Amazon, we took a regression approach using 13 years of malaria incidence data from 795 municipalities, along with remotely sensed data on forest cover and loss. And the regression is basically what is malaria incidence as a function of forest cover, forest loss, and an error term. But of course, as you've probably already recognized, there's some potential pitfalls to this type of approach. There could be unobserved and omitted variables that are truly driving the relationship, and we could either fool ourselves into seeing something that's not really there or obscure the true relationship. And because we hypothesize simultaneous causation, how are we going to disentangle these two directions of effects? How are we going to know which is causing which? And here, to address these two issues, we borrowed some approaches from the econometrics toolbox. These are really a set of tools that have been developed for systems like this one, where it's impossible or unethical to do randomized controlled trials. So you can't do an experiment. So we can take statistical approaches that do the best possible, closest possible approximation to a randomized controlled trial. And so the first tool we're doing, if we want to avoid omitted variables, we should include covariates that we know are going to affect either forest loss or malaria. So things like climate, urbanization, and wealth. The next thing we can do is we can be even more rigorous, and we can control for things that are unobserved, but we expect might confound the relationship. So we're now including dummy variables, or in econometrics, what are called fixed effects, for each municipality in each year. So we're now just looking at the deviations from the average for a given municipality in a given year in forest loss and malaria, and asking whether those deviations are associated with each other. And then finally, because we hypothesize this feedback, we can statistically test for whether there's endogeneity in the model, which could suggest a feedback, by looking at this error term and asking whether the error term is statistically correlated with the malaria incidence, which would indicate a potential feedback. And if there is support for this feedback, we can then use what's called an instrumental variable to try to just look at one direction of the effect. So in this case, we're using an instrumental variable of aerosol pollution, which is highly related to deforestation because it increases as you increase deforestation, but it's not otherwise expected to be independently related to malaria. So the idea here is if aerosol pollution appears to have a positive effect on malaria, that's really a signal of deforestation affecting malaria. Then we can reverse this whole econometrics approach and ask the opposite question. How is malaria influencing deforestation? Again, using these fixed effects and the instrumental variable. And in this case, our instrumental variable for malaria is the temperature suitability for transmission, based on that mechanistic model that I presented in the first part. So because the malaria transmission season is actually the dry season in the Amazon, how many days during the dry season were the temperatures between 24 and 26 degrees Celsius, which we've already seen are highly suitable temperatures for malaria. So we expect that to be closely related to malaria, but not independently affecting deforestation. So given all this background on the statistical methods, the results now are actually very simple to interpret. What we found, first of all, is that deforestation strongly increases malaria. This is our full statistical model, including fixed effects and our instrumental variable for forest loss. And we include that instrumental variable because there's significant evidence statistically that that variable is required. So in order to reduce that endogeneity, we needed to use the instrumental variable. What we find is that this effect in magnitude is actually fairly large. An estimated about 6.4 new malaria cases are expected to occur for every square kilometer of forest cleared. Now we can also find the opposing effects. So we see evidence statistically that malaria actually reduces deforestation, again, using this fixed effects model with an instrumental variable for malaria. Again, we saw statistical support for that feedback, which motivated our use of the instrumental variable. And again, we see a fairly substantial effect size. 0.072 fewer square kilometers are cleared per malaria case. Now, testing that second hypothesis, does this relationship actually vary with the stage of land use transition? We're now going to break our data set up into that interior versus outer region and do the same regressions and just compare the effect sizes. And what we see is that when we look at the frontier region of the Amazon, where these frontier clearings are occurring, we see about twice as strong of an effect of forest loss on malaria. And we see a stronger effect of malaria on forest loss. So this bidirectional feedback is much stronger in the interior of the Amazon in this frontier and subsistence agriculture stage. By contrast, in the outer region of the Amazon, there's no significant effect of forest clearing on malaria. It's a very small positive statistical estimate, but there's almost no effect. And again, that's because there's very little forest left. So clearing the forest doesn't really increase forest edge anymore at this point. And as you would expect, since there's not much malaria in this region, there's also not a significant evidence for a feedback of malaria incidence on forest clearing. So that is really a frontier malaria story that's going on in the interior of the Amazon. So to summarize, we see evidence for a bidirectional feedback between deforestation and malaria, where within the forest, where there's lots of forest cover remaining, deforestation promotes malaria by creating forest edge and increasing human contact with the vectors that live in the forest edge. We also find that deforestation declines with the highest malaria incidence, and we're still a little bit unsure of exactly how that mechanism works, but it may be related to either people being too sick to work or settlements being abandoned in places with high malaria incidence. And we find that these relationships are strongest in the frontier regions of the Amazon, so deeper into the Amazon where less of the forest has already been lost. Just to add sort of a current event note on this story, under President Bolsonaro, he's been really aggressively promoting environmental policy that allows deforestation in the Amazon. And deforestation was up by 30% last year to almost 10,000 square kilometers. From this model, we would project that 10,000 square kilometers of additional deforestation would cause about 62,000 new malaria cases. So what that tells us is it allows us to really put a human health number on these environmental impacts, which we hope could be important for policymakers should they choose to use this type of evidence in their decision making. So to wrap up, how is global change altering infectious disease? In the first part, we talked about climate change. And what I found is that climate change is really expected to drive shifts in transmission rather than widespread increases in most diseases. We also find that despite all the complexity and all the nonlinearity, these changes are really highly predictable from these nonlinear models, including spatial relationships of where malaria incidence is peaking and other diseases, but also temporal relationships, especially if we have access to information about susceptibility of the host populations. For land use change, we find that land use change does affect transmission, and it's mediated by vector ecology. So for the case of Amazon malaria, because it's a forest edge vector, we see this strong association between the types of land use change that create forest edge and malaria incidence. And we also see evidence that human behavior can actually respond to disease, because diseases are embedded in these socio-ecological systems with socio-ecological feedbacks between human land use decisions, vector ecology, disease transmission, and human decisions responding to that disease. And what we found more broadly, just recently, actually in 2019, I worked with a class of undergraduates to do this review paper where we asked, what has been the impact of vector-borne disease across the arc of human history? And we took this type of framework, thinking about the human-made environment, so that's shown in yellow here, things like agriculture and urbanization, affecting vector ecology, which affects disease burden, which in turn affects the social responses to diseases. And we focus this review on 4 key pathogens that have been really important throughout human history: plague, malaria, yellow fever, and trypanosomiasis. And what was really fascinating is we found this whole range of different impacts that vector-borne diseases have had on human history. Of course, we know about the Black Death and the major demographic impacts those have had, but we also found that yellow fever has mediated the outcome of colonial excursions in Latin America. So for example, in some cases, it helped the Spanish to defend colonies that they had already colonized because they had differential resistance to this disease. We also found that malaria was really instrumental in determining the outcome of the American Revolution, because the Continental troops had previous malaria exposure and had some resistance, and they were able to draw the British Army into malaria-prone regions and really decimate their troops that way. And one of the most profound things that we found in this review was the extent to which existing systems of oppression and inequality really fed into the effects of vector-borne disease. So just like we see today, there's really unequal impacts of vector-borne disease that are really mediated by unequal social systems. And we found that this has a really big impact on vector-borne disease dynamics. So to conclude, socioecological feedbacks have nonlinear and yet highly predictable effects on infectious disease dynamics if we can take those feedbacks into account and really use models that incorporate data as well as mechanistic understandings of disease systems. So thank you very much for your attention, and I'd be happy to take any questions.
So we have the first question from Nicholas, and he said, do you predict that malaria rates would increase following more sustainable forestry operations as well?
I think that really depends on how that forestry is managed, particularly in the Amazon. You know, if it could potentially, if you were creating lots of forest edge and you had people living, especially in informal housing that wasn't sealed and didn't have good window screens and good infrastructure and good public health infrastructure. But you could also envision that sustainable forestry practices could help to prevent malaria by preventing the widespread sort of formation of forest edges, like what I'm showing in this really horrible picture here. So I think it really depends on the nuances of how that land is being managed.
Thanks, Erin. We have another question coming in from P. Mayer. Are the R0 optima a function of enzyme kinetics associated with some aspect of the life cycle process of the vector? And if so, can enzyme kinetics improve the climate-driven models?
Yeah, so R0 is an emergent property that emerges from a lot of different traits of vectors and parasites, which include many that are dependent on enzyme kinetics. So I think if you wanted to get more mechanistic, and especially if you wanted to be able to predict these a priori without actually doing all the lab experiments, I think if you had a really good understanding of the biochemical processes underlying development rates and reproduction rates and survival, then you might be able to take the temperature dependence of enzyme kinetics and use that to predict how these emergent traits and transmission would eventually scale with temperature. So I think it is a really promising area for more mechanistic, molecular-based approaches to these questions. But I don't think you could directly apply enzyme kinetics and go ahead from there and predict global changes in disease transmission. I think there's several steps in between.
All right, thanks, Erin. We have another question from Marie Toza. You touched on this a little bit, but could you expand on whether you think it's possible for mosquitoes to co-evolve with climate change?
That's a really interesting and important question, something that we've been trying to get at with some new empirical work that we're working on in my lab and with some collaborators. The thing that's most limiting to transmission at high temperatures, it's not that you don't have any mosquitoes where R0 is starting to decline, it's that the mosquitoes don't live long enough. Because for a mosquito to get infected, it has to take a blood meal, get parasite in that blood meal, and then live through that incubation period to where it becomes infectious. And then the longer it lives from there, the more people it can go on to bite and transmit to. And so in order for transmission to continue under warmer temperatures, you would need lifespans at warm temperatures to increase, because that's really that's the trait that has the lowest thermal optima across all the species we looked at. So what's interesting is that evolution could affect the mosquito and the parasite differently, because the mosquito might not need to live longer at warm temperatures to optimize its fitness. It might be able to just develop and reproduce very quickly, but the parasite needs the mosquito to live longer to be able to be transmitted. So that's sort of a hypothesis. What we don't know is what are the genetic mechanisms underlying thermal tolerance, and how much variation is there in thermal tolerance across populations? We know a little bit more across species, but across populations, how much local adaptation to temperature is there? And that's something that a graduate student, Lisa Cooper, in my lab is trying to work on with the Western treehole mosquito across the west coast of North America, which is it's a system where there's a huge climate gradient in its range. So we're interested in trying to use that to understand what are the genes involved in temperature adaptation and how much variation is there across this mosquito's range. Were there any other questions?
I'll ask a question. That was excellent. Thank you.
Thank you.
So have you thought about what kinds of anti-malarial interactions between medical services that are provided at these frontiers and how that might interact or not interact with the patterns that you see? Because certainly there may be public health data on that, but who knows?
I don't know too much about access to anti-malarials in this region. One piece of research that I didn't talk about today, because it's really, really new, but I have another student who's working on effects of gold mining on malaria, specifically like small scale alluvial gold mining, which is usually illegal in a lot of the Amazon. But it's another situation where we've seen really strong associations, at least spatially, like places with lots of gold mining have lots of malaria. And there's been implications locally at different places that gold mining promotes malaria, but we haven't been able to get like a really rigorous, broadly applying estimate yet. And so the student, Marissa Childs, has been using econometric approaches similar to what I showed for deforestation, and showed that there are big effects of local gold production on malaria. On the order of, a kilogram of additional gold production is expected to cause about 20 new malaria cases. But what's really interesting is these two malaria parasites, the Vivax malaria parasite, can actually hang out in the liver. So once you recover from your acute infection, you become chronically infected, and you can have this recrudescence where you get sick all over again months or years after the initial infection. So what we see with Vivax is not only local scale immediate effects, but actually spillover effects of gold mining. So we can actually detect the effect of gold mining on malaria up to 250 kilometers away from the place where gold mining is actually happening, because people are basically getting infected during gold mining and then moving on and bringing the malaria parasite with them. So that's an area where, treatment that could clear the malaria parasite earlier might actually be able to prevent some of that. And, certainly it would reduce the burden of, the actual health burden that people are experiencing if they have access to anti-malarial drugs.
Thanks, Erin. We have a couple more questions. One from Donna Skurlock, who asks if using clinical malaria rates to humans in the Brazil deforestation scenario, as an example, are diagnostic tools similar, or are more rural sites using clinical criteria alone versus like clinical plus addition of laboratory for more exact diagnosis in order to compare?
That's a great question. And I don't know the details of how malaria diagnostics vary across the region. You could imagine certainly that there might be less diagnostic capacity in more rural places. But if that were the case, I think that would probably make the results a little more conservative, because that would mean we're probably underestimating malaria a little bit. But in the most malaria-ridden areas.
Thanks, Erin. And I think we have time for one more question.
Okay.
We have a question: if historic DDT use, non-use, or mosquito tolerance to DDT has influenced the increase in malaria in Brazil in concert with deforestation?
That's another good question. I mean, again, I don't know the history of DDT use in the Amazon specifically, but more generally across the Americas, DDT was like this wonder chemical when it was discovered, and it was used everywhere. And it was a really critical tool in eliminating malaria in the US, where we had really, a substantial malaria problem prior to World War II. And there were huge efforts, including a lot of DDT usage to try to eliminate malaria. And it's since kind of been taken off the table because it's so environmentally destructive. But the problem is that the remaining tools are not always as effective. And so, yeah, I don't know exactly how that relates to previous DDT usage, but in general, there was a big push for vector control throughout the Americas, starting around World War II and then going into the 1970s, which then kind of became a victim of its own success. We especially see this for Aedes aegypti, the dengue vector, where its range had really contracted, and vector control was really effective at eliminating the range of Aedes aegypti. And we saw very little yellow fever and dengue transmission during that period. But then, we lost focus. We weren't ever able to actually eradicate or eliminate these diseases or these mosquitoes. And after the focus and the money dried up, the vectors came back and the transmission problem came back. Now, malaria control has been pretty successful in most of of Latin America, with the few remaining places that have malaria, including the Amazon, including some of it coming out of Venezuela, and some of it actually coming out of gold mining areas in places like Costa Rica, where they've otherwise eliminated malaria. Of course, the really big mosquito-borne disease problem in most of Latin America now are diseases transmitted by Aedes aegypti, like dengue, Zika, and chikungunya. And that's where the big control efforts are really focused today. Yeah, so I see someone's asking about gene drive. And that's one strategy that's come out of this major problem. And there's good and bad things about gene drive. The good thing is that it's really, really specific. So it's not going to have the off-target effects that something like DDT does. The bad thing is a lot of forms of gene drive, depending on how they're designed, they can either be self-limiting or self-perpetuating. And the self-limiting ones, that they're safer in a sense because they have an off switch, but they also just require so much effort. You have to continue to introduce, sterile insects, for example, into a population. And of course, there's risks of gene drive in the sense that those genes can spill over if there ends up being hybridization with wildlife, or wild insect species, and they can simply not always be effective depending on the situation. So I think they're a potential tool in the toolbox that should be considered, but they're certainly not going to be a silver bullet solution that's going to take away the problem of malaria or mosquito-borne disease.
I think that's all the time we have for questions. I'd like to thank Dr. Mordecai again for coming and giving this talk, and remind everyone our talk next week will be by Abba Gumel from Arizona State University, looking at how could face masks curtail the post-lockdown resurgence of COVID-19 in the US. Thank you.
Thank you.
Erin, thanks so much. That was really great. My daughter's making strange noises here.
Thank you.
Thanks a lot, Erin. That was
Thank you.
excellent.